The changes in the osmotic concentration of the glomerular filtrate as it passes through the renal tubule. The glomerulus is the filtering unit of the kidney, made up of a bundle of capillaries, and is located between two resistant vessels. On this photo here, or on this diagram, you can see the glomerular capsule, which is also known as Bowman's capsule. You can see the glomerular ultrafiltrate, the efferent arteriole, where the substances come out, the afferent arteriole, where the substances go in, and then in green are the protein, if you can see those little green dots, and the purple is the other solutes. What is glomerular filtration? Glomerular filtration is the first step in creating urine. It is caused by a hydrostatic and osmotic pressure. This process is used by the kidneys in order to transfer excess waste and fluids out of the bloodstream and into the urine collecting tubules. What happens during glomerular filtration? So just a little side note here, glomerular filtration process is also called renal ultrafiltration. And what happens during this process is blood pressure forces water and solutes from the capillaries into the glomerular capsule by a way of a filtration membrane. The filtration membrane blocks cells and large proteins from passing, leaving them in the bloodstream, all while allowing water and small solutes to pass further into the nephron. So a way that I remember that process is, if you're cooking pasta, once the pasta is done, normally you will use a strainer to drain the water out of the pot where the pasta is. And if you are familiar with pouring pasta into a strainer, you know that all the water goes down into the little holes and the pasta stays in the strainer. That's just kind of similar to what the filtration membrane does. Glomerular filtration rate. The abbreviation for glomerular filtration rate is GFR, and it is the volume of fluid filtered through the body's glomeruli per unit time. Hydrostatic and osmotic pressure. Hydrostatic pressure is the force of pressure exerted from pressure of the blood vessel. Osmotic pressure is the pulling force exerted by albumins. And that's kind of what we're going to focus on here in this presentation, is the osmotic concentration and pressure. Osmotic concentration changes. When filtrate passes through the nephron, while the water and ions are reabsorbed, the osmotic concentration changes. The filtrate is 300 osmoles of solute per liter when going into the proximal convoluted tubule. When the filtrate is leaving the proximal tubule, it still remains 300 osmoles of solute per liter. However, when the water is leaving through the descending loop of Henle, the osmotic concentration will then lower to 1200 osmoles of solute per liter. The osmotic concentration then decreases again to 100 to 200 osmoles per liter. Based on hormonal stimulus, the water and ions may be reabsorbed. The final osmotic concentration is dependent on the final collecting tubules and ducts being permeable to water.